In lab, we talked a little bit about meristem and meristematic tissue, which is undifferentiated and therefore the same kind of thing as the stem cells in animals, capable of further specialization into eventually fully differentiated states where they carry out their final orders in the integrated multicellular organism. In order to become specialized, like a leaf cell or a kidney cell, you have to exaggerate one tiny part of the cell's overall potential. Remember that each cell has the same DNA, the same information required to be anything within the body of the multicellular organism. And yet it doesn't. Nor would it be a good idea for it to try to do everything. The multicellular organism works out as better than a cluster of unicells because of the specialization of cells to particular roles. And in the process of specialization, you have to give up the ability to do other things, and also, usually, the ability to undergo cell divisions. We can safely characterize meristematic cells as having no physiological role. They don't transport nutrients or photosynthesize or eliminate waste, but they do undergo cell divisions and thus give rise to other cells that may become specialized. And just the fact that these cells are the ones undergoing cell divisions means that they're responsible for all of plant growth. If most of the cells in a plant's body, or your body for that matter, are the sterile workers of an ant colony, the meristem cells are the reproductives, the queen ants in a multi-queen colony, capable of breeding and producing more sterile workers in various castes. The vascular cambium is one such meristematic tissue. This is the thin layer of cells between the xylem and phloem and the vascular bundles of eudicots, like medicago, and between the xylem and phloem and woody stems, like what we saw in lab with quercus or tilia. The two main kinds of cells produced by the vascular cambium are the two kinds of vascular cells, phloem and xylem, actually protoxylem cells, because they're just the framework and scaffolding upon which xylem is constructed. Once the woody tubular structure is complete, the actual cells are destroyed, leaving just the hollow tubes to serve as water ducts. Vascular cambium is one form of lateral meristem on the sides of plants, and this layer is responsible for all secondary growth, that is, growth in thickness or the girth of the stem. For example, as the main stalk of a skinny sapling grows into the thick trunk of a mature tree. Effectively, all of that growth, all of that wood, is produced through the deposition of layer after layer of xylem by the vascular cambium, leaving behind a tissue comprised of tightly packed, sturdy tubes. The fact that wood is strong and a good building material is a reminder to us of xylem's other important role in the growth of plants. It's the tissue that holds up an enormously heavy canopy of limbs, branches, and leaves. The phloem is also produced by the same vascular cambium, albeit at a slower pace. Phloem never takes on the role of supporting the plant's weight, and so there's no motivation for the plant to produce more of it than what's needed to transport the sugars and hormones throughout the plant. Compared to the xylem, there's only a thin skin of phloem in a tree trunk sitting just outside of the vascular cambium. Yes, the layers of phloem are deposited in the outward direction from the vascular cambium. The phloem is the tissue through which sugars are transported, and with it being on the outside, it's easy to see how girdling a tree, like when a rodent eats through the bark all the way down to the wood in a contiguous band all the way around the trunk. This effectively cuts off the flow of sugars to the root system, which then dies, resulting in the death of the whole tree. Another fun fact about vascular cambium: the technique of grafting, putting a part of plant B onto plant A, creating a chimeric Frankenplant in the process, requires that the vascular cambia of the two plants match up in a way that allows them to fuse into a single meristem. Once this happens, the two plants can continue to live, exchanging water and nutrients across their cells, as though they were cells of a single plant. Grafting is a widely used technique to clone. For example, If you bred a tree that produces particularly excellent fruit, whose quality is a lucky result of the tree's unique combination of genes, you couldn't reliably get more plants with similar fruit quality just by planting the seeds produced by your plant. That wouldn't work because those seeds are the product of recombination, and through meiosis and fertilization, the perfect gene combination of your wonderful tree would be ruined through mixing, and so the resulting offspring plants would not produce fruit of the same quality. Grafting is a form of cloning, which is asexual reproduction because it circumvents recombination. Your wonderful fruit tree would probably start off one of a kind in an orchard with a bunch of other trees that produce mediocre fruit. By cutting the mediocre trees down to stumps, letting them regrow, and then grafting branches from the wonderful tree onto the regrowth, soon you'll have a whole orchard of fruit trees that may be all different at their bases, but the fruiting part of the trees will all be cloned from and therefore carrying the same gene combination of the original tree that makes wonderful fruit. This same technique can be used to produce novelty trees with more than one type of fruit being produced on a single tree. I planted this avocado tree in my front yard, and after it grew, I decided that its fruit was just lousy. So I stumped it, and I grafted two good avocados onto the regrowth, and now I have reed avocados and fuerte avocados growing on a single tree. This technique does not work across different species. You can't graft avocado onto a lemon tree, but you could graft tangerine, which is also citrus, onto the lemon tree. Now, besides vascular cambium, the other meristem that we'll talk about is the meristem that you find at the tips of growing vegetative branches and the tips of growing roots. Generally, the class of meristem occurring at the tips of structures is called apical meristem. Literally, meristem occurring at the tips of structures, in contrast to the lateral forms of meristems like vascular cambium. Apical meristems are responsible for primary growth, that is, growth in length of shoots and roots, and we distinguish between shoot apical meristems and root apical meristems, SAMs and RAMs, to the people that use these terms a lot. Primary growth occurs through two basic mechanisms: an increase in cell number occurring through the cell divisions of the apical meristem, and also elongation of the cells, which are minuscule squat little things when they're first produced by the meristem, but then stretch out into long cell bodies before finally maturing with the deposition of a rigid cell wall that sets the final cell size. This elongation is something we come back to later. As the cells of the shoot apical meristem continue to divide, they leave behind them the cells of all parts of the stem, including the leaves, to swell up, elongate, and become specialized. This actually includes rings of xylem and phloem, with a ring of vascular cambium between them. In fact, the xylem and phloem produced by division off the apical meristem is called primary xylem or phloem, while all the layers produced by division off the vascular cambium are secondary. So, primary growth occurs through a lengthening of a growing tip. Secondary growth occurs through a thickening of the stem or root. If these two forms of growth was all there was, you'd be able to make plants that grow up as single stalks, with roots that grow out as single thickening cords that extend but don't branch. In order for a plant to look like a plant, or at least a plant like one we would find familiar, it would have to have some element of branching, either with one branch or root splitting into two, or with side branches growing away from the main branch. So the third distinct form of plant growth, after primary and secondary growth, is that of lateral branching, which is basically the creation of new stems and roots to the sides of existing stems and roots. Each new lateral stem or root has its own apical meristem undergoing primary growth, as well as its own lateral meristem causing secondary growth. This phenomenon of branching occurs a little differently in shoots versus roots. In the above ground part of plants, and here I'm really talking about most non-monocot flowering plants, the apical meristem of the shoot grows upwards and outwards, leaving behind it cells that differentiate and mature into many different tissues and organs. One of the things produced is an axillary bud, a tiny embryonic shoot that sits inside the little wedge where a leaf attaches to the main stalk. We call this the leaf scar because after the leaf dies and falls off, you can see the scar where it was attached. And this tiny embryonic shoot is sitting there waiting for its chance at glory. Under the right signals, the tiny primordium of a bud becomes active, swelling up and then growing into a lateral branch. Manipulating the signal to either activate or not activate is how the plant controls its basic growth form. Is the plant going to be a low-growing shrub with lots of lateral branching, or a tall tree with no branching at all in the main part of its trunk? The developmental switches that determine plant shape exert their control by manipulating the amount of branching that occurs in the shoot. Now, I've mentioned before that the plants of the Carboniferous would have seemed somewhat alien to us because they were basically tree-sized versions of club mosses and horsetails and ferns. Even when we get to the Permian with the new appearance of seed-bearing trees, these weird plants continued to be abundant. Okay, we talk about these plants as weird or alien because of their appearance. The fossils of Lepidodendron and Watiza and Saronius and most of the other famous trees of the Paleozoic appear weird to us today because of how they lacked branching. For me, visualizing a world in which plants don't have branches is part of the fun of recreating the narrative of life in ancient times. In the next lecture, we'll focus on the basic mechanism underlying the operation of the two vascular tissues, xylem and phloem.